Assalamu alaikum and welcome to Ordinary Days. In today's video, we are going to discuss chapter number 46, which is somatic sensations. So, what is actually somatic sensations? These somatic sensations are actually collect the sensory information from all over the body. So, somatic sensations are sensory sensations collected from all over the body. And classification, we can classify the somatic sensations as mechanoreceptive sensation, thermoreceptive sensation, and pain or nociceptive sensations. Mechanoreceptive sensation, they can be further divided into tactile or position sensation. Thermoreceptive can be divided into heat and cold, like hotness and coldness, and pain or nociceptive sensations are produced when there is any damage to the tissues. So now, in this chapter number 46, we are going to discuss mainly the mechanoreceptive information, that is tactile and position one. And in tactile, we get touch, pressure, vibration, tickle, etc. Whereas in position one, we get whether this position is static position, dynamic position, and the rate of movement. So classification is mechano, thermo, and nociceptive. Now we can classify the sensations on the basis of their location. Like they can be classified exteroceptive, that are received from surface of the body, and proprioceptive, which are coming from inside the body. The proprioceptive information, they include visceral sensation and deep sensation. Visceral sensation are coming from the internal organs, and deep sensation, they are coming from muscles, from bones, from the fascia, etc. And these sensation are PVP sensation. They are pressure, vibration, and pain sensations. So this is how we classify them on the basis of their location, and this is how we classify them on the basis of their sensation. Now, how do we detect the tactile sensation? I told you that this chapter is going to focus on the tactile sensations and the position sensations. So first of all, we are going to deal with the detection of the tactile sensations with their receptors and how they are transmitted. So let's discuss tactile sensations. So tactile sensations, tactile means touch. So they can be touch, they can be pressure, they can be vibration. Touch is detected by tactile receptors. Pressure is detected via deformation of the deep tissues, and vibration by repetitive sensory signals. And tactile receptors, the touch receptors, there are 6 types of tactile receptors. We'll discuss each and every receptor in detail, and we'll discuss which one adapts faster, which one adapts slower, which will help us to classify whether they are detecting slow sensations or fast ones, etc. So out of the 6 receptors, we have free nerve endings. We have Meissner corpuscle, Merkel disc, hair and organs, Ruffini nerve endings, and Pacinian corpuscles. First of all, we are going to discuss the free nerve endings. They detect touch and pressure sensations. For example, they are present in the cornea of the eye, and for example, this is the cornea. So whenever you're going to touch the cornea, you're going to press the cornea, all of those sensations will be felt because cornea contains the free nerve endings. So in this diagram of the Guyton, you can see these free nerve endings. How are they? Like they are not encapsulated or anything, but they are freely present. And most common location is like the cornea of the eye, but mostly they are present in all over the body, and they help in touch and pressure detection. Next one is the Meissner corpuscle. They have increased sensitivity. They are elongated encapsulated nerve ending. You can see it is elongated and it is having this capsule around it, so it is encapsulated nerve ending. And the type of fiber that is helping in its transmission, it's a beta fiber, which is a large fiber, and it is myelinated. So it is a encapsulated elongated nerve ending with a beta fibers, which are large myelinated fibers. And where are the Meissner corpuscle present? They are present in lips, fingertips, non-hairy parts, and it adapts in a fraction of second. So it has extremely fast adaptation. What does fast adaptation help in? It helps us in detecting the movement of the object, any rapid movement, and sensitive to low frequency vibrations. Any low frequency vibration, any rapid movement, it will be detected by these Meissner corpuscles, which are rapidly adapting type of receptors, tactile receptors. And over here, these are the Meissner corpuscle, elongated encapsulated nerve ending with A beta large myelinated nerve fibers. Then we have the Merkel's disc, which are expanded tip tactile receptors. In this one, in the major difference between Meissner corpuscle and Merkel disc is, in this one, the whole nerve fiber and its branches, they are encapsulated by one capsule, whereas this Merkel disc, each fiber is encapsulated by another capsule. So it is an expanded tip tactile receptor. All of its tips are encapsulated and expanded. It can be present in hairy and non-hairy parts, and the major difference between the Meissner corpuscle and Merkel disc is that it, first of all, it gives a very strong signal, and then after that it transmits a weaker signal. So it means that it adapts slowly and progressively and gives out steady state signals. So when it adapts slowly, it means that it will help us to determine any continuous touch of the object. So if you're pressing the finger, the initial press, it is detected by the Meissner corpuscle, but the elongated continued press, it is detected by these Merkel discs, which are adapting slowly. And both the Meissner corpuscle and Merkel disc, they help in localization of the sensation and help us in the texture perception. And also these, the area of our skin where this Merkel disc is present, it forms a elevated thing which is known as a touch dome. Also, the sensations that it is transmitting, they are also carried by this A beta fibers, which are large diameter myelinated nerve fibers. Now we have the hair end organs. The hair end organ, it is actually hair plus the basal nerve fiber. So if we look at the diagram, so this is the diagram, and in this diagram you can see that the hair end organ is actually the hair, the distal part of the hair, or the proximal part of the hair, and the nerve fiber. It is also carried by this A beta type of large myelinated fiber. Now it is hair plus the basal nerve fiber, and it is a very rapidly adapting one. So if it rapidly adapts, so it means that it is going to detect any initial contact or movement of the object. So this is how it occurs. Any random touch, any random contact, it is going to be detected by this hair end organ. Then we have two others, Ruffini and Pacinian corpuscle. Now this Ruffini nerve ending, it is present in the deeper layer of the skin. It is multi-branched encapsulated nerve ending, very similar to the Meissner corpuscle. This is the Ruffini ending, which is multi-branched. It is having multiple branches and it is an encapsulated nerve ending. But it is present deeper in the skin, whereas Meissner corpuscle is present superficially. So now this Ruffini nerve ending, it adapts very slowly. So it is involved in continuous deformation. Anything that adapts slowly, it means that it is going to detect any continuous signal. Continuous deformation, heavy prolonged touch or pressure signals. Now we have the Pacinian corpuscles. The Pacinian corpuscles, they are also present. They are present immediately beneath the skin. As the name suggests, P for Pacinian, P for pressure. So they detect vibration, pressure and mechanical state of the tissue. They detect rapid local compressions or local pressure sensations, and they adapt very fastly, that is in a few hundredth of a second. So whenever they are going to adapt very fastly, it means that they are going to detect any rapid type of sensation, whereas the ones which adapt slowly, they detect any continuous type of sensation. So one which adapt fastly, I have a mnemonic for that, that is PMH. P is for Pacinian corpuscle, M is for Meissner corpuscle, and H is for hair end organs. And adapt slowly, they are FREM. From the small form of friend, we have the FREM. F is free nerve ending, R is the Ruffini nerve ending, and M is the, if you trace it backwards, MER, so it's Merkel disc. So this is how you can remember the ones which adapt fast and the ones which adapt slow. The ones which adapt slow, they are involved in continuous deformation, pressure sensation, prolonged touch. Whereas the ones which adapt fast, they are involved in detection of any change in movement, any local compression, rapid compression, all of that thing is by fastly adapting ones. Now we are done with the tactile receptors. Now let's discuss how the transmission of the tactile sensation takes place. As we have already discussed, Meissner corpuscle, Merkel disc, Ruffini nerve ending, Pacinian corpuscle, um, they are all they all send this sensation by A beta fibers, which are large diameter fibers. So the intensity of traveling is 30 to 70 meters per second, whereas the free nerve endings, which are like the ones that have no encapsulation, they are transmitted by small A delta type of fibers, which transmit in 5 to 30 meters per second, or by C type of fibers, which are non-myelinated, which travel at 2 meters per second. Now we can talk about the A beta fibers, which are large diameter fibers. Now they help in, and they are also myelinated, so help in localization, gradation of intensity, and rapid changes in sensory signals. Localization, gradation, rapid changes. Whereas the C fibers, which are involved in crude touch, they are slower because they are non-myelinated. They are very small nerve fibers, and they detect pressure or poorly localized touch and tickle. So that in that we are done with all the transmission of the tactile sensation, their receptors, how they are transmitted. In the next video, we will start with detection of vibration.